Welcome to the Spring 2021 Core Facilities Seminar. This is the last seminar of this series, and today we will be featuring the Genomics Resource Laboratory. I am Lisa Korpieski, the Director of Communications and Marketing here at the Institute for Applied Life Sciences. In today's seminar, we will hear from Ravi Ranjan, the Director of Genomics. He will tell us about the suite of services available to address your high-throughput next-generation sequencing needs. Our guest speaker is Elizabeth Rullo from 10x Genomics. The title of her talk is Uncover the Hidden Complexities of Biology with Single-Cell Solutions. We hope that with these bi-weekly seminars, you will discover what great resources that the centralized UMass Amherst core facilities offers to our campus community, the New England region, and beyond. Next, just a few housekeeping items. The seminar is being recorded, as you just heard. If you miss any part of the seminar or would like to forward it to someone who could not attend, there will be a replay of this and all previous seminars in the series on our website. We will put a link in the chat for you. I recommend you set your view mode to speaker. Please stay muted during the talks. We will save the Q&A till the end of the presentation. During that time, I welcome you to just unmute yourself and ask your question. And now I would like to introduce you to Andrew Bernard, Director of our centralized UMass Amherst core facilities. Andrew?
Thank you, Lisa. Good afternoon, everyone. Thank you for joining us today for the 9th and last seminar of the Spring 21 Core Facility Seminar Series. We have some additional programming during the summer that I look forward to sharing with you and hearing from you what else you'd like to see from us. As Lisa mentioned, my name is Andrew Bernard, and I'm the Director of Centralized Core Facilities here at UMass Amherst. Our state-of-the-art labs, instrumentation, equipment, and world-class experts are helping researchers drive discovery and pursue scientific endeavors across all disciplines. The cores are open to anyone from students through senior scientists, regardless of your affiliation, including researchers from other academic institutions and commercial operations. To help access the cores, there are several funding opportunities. For our IELTS investigators, core credits and central seed funding is available. For those external to UMass, the Mass Innovation Voucher Program subsidizes usage for small companies in Massachusetts at up to 75%. To date, the program has awarded more than 600 vouchers, representing over $5 million in total project costs across the 5 UMass campuses to Massachusetts businesses. We aim to be your partner by expanding your research capabilities and productivity. You can feel free to reach out to me in the chat or during the Q&A, or reach out at any time to any of the core facility directors if you have questions about our capabilities or any other aspect of working with the cores. It's now my pleasure to introduce Dr. Ravi Ranjan, Director of the Genomics Resource Lab. Ravi joined UMass in 2017 from UIC. After about a month on the job, the facility experienced a full-on flood that in hindsight was probably really indicative of the myriad challenges Ravi would be facing as a core director. More importantly, it was really representative of how Ravi stands up and responds to challenges. When he's not facing floods, Ravi provides next-gen sequencing services from sample prep to generation of high throughput sequencing data. He's here and available for everything from instrument training to analyzing the data and helping you with protocol development. He is incredibly knowledgeable and resourceful in finding resources both inside and outside of UMass, so we hope you will engage. Ravi also stays engaged on the national stage with ABRF and other initiatives to make sure that we have the best knowledge here at UMass. It's why one of the reasons we're talking to 10x today. And he makes sure that all of our faculty have access to those resources as well. Over the last year, Ravi stepped up in a major way to lead efforts in establishing the IELTS Clinical Testing Center, both in acquisition of equipment and setting up all that new equipment, but also in development and validation of the assays that have gone on to test tens of thousands of UMass students and staff and the community at large. He's an asset to UMass. We are grateful and privileged to have him, and I look forward to hearing him give you a little bit of spiel about the Genomics core before we have some communication with 10x today too. So. With that, I'll turn it over to Ravi and be here for questions if there are any. Thank you.
Thank you, Andrew and Lisa for the introduction. And I really appreciate all the thoughts into that you have said for my role for running the genomics facility. So thank you very much. Like Andrew mentioned, I'm supervising the genomics facility at UMass Amherst, and we provide a suite of services for next-generation sequencing services. We provide nucleic acid isolation, qPCR assays, library preps. In terms of NGS, we support microbial genomics, environmental genomics, mammalian system, NGS, the plant genomics, and any other systems that we work on. The general workflow of the NGS is sample collection, experimental setup, sample collection, depending on the assay. If it is DNA genomics, then it is DNA. If it is transcriptome, then it is RNA. Then we do DNA RNA extraction. We process the samples for sequencing. We call that process as library prep. We have two Illumina sequencers, the MiSeq and the NextSeq. Depending on the application, we use either of the instrument, or we also help in outsourcing the sequencing projects on a larger commercial platform such as NovaSeq. We also support with limited data analysis. We also assist with results interpretation, and we provide publication quality figures as well as help with grant applications. In terms of our lab, we have specific assays that we are very confident in running. One is for the microbial genomics. We can assist with the metagenomics, that is the amplicon sequencing, as well as the short-term metagenome sequencing. We also have assays developed in the lab for metatranscriptome, and I can talk about the cost, sample processing time at a later discussion. For the plant or mammalian systems, we have whole genome assays, whole exome sequencing, and we also support a good amount of bulk RNA-seq assays. But nonetheless, we are also inclined to work on other projects. We also finished small RNA sequencing projects. We are now discussing to work with one of the investigators for the 3' gene expression RNA transcriptomics. We also have, we are also interested in any other epigenetics sequencing projects or ChIP-seq. So feel free to reach out, but these are the ones that we have established in the lab. We are also working with one of the investigators for CRISPR sequencing projects, and it's work in progress. So it is not just the custom projects that we offer, but we are also open to any custom projects, and whatever related to genomics, we can help you, or we'll try to help you as best as we can. Other services, like I mentioned here, we prepare libraries, whole genome, shotgun metagenomics, transcriptomics, small RNA-seq, and we have a good resource for DNA and RNA isolation. And now we are working on a single-cell genomics. We have the FluidArm system, but depending on the needs and other resources, we are planning on other modalities. And so we have invited 10x Genomics to discuss about their system and how it can be useful for the research community. Other services, we provide instrumentation training. This is for fees, fee for service. So, but we try to module that training in such a way that the data can be used by the students to generate data. We also assist with NGS project consultation, any technical assistance or troubleshooting. We provide documentation for grants. We prepare budgets. We provide letter of support. I provide BioSketch if I'm a co-I or as needed. And we also provide documentation for manuscripts. Recently, I was fortunate to be included as a co-I in the grant that is funded now. So again, it is, and that in my role is, it's not like we offer standard services. That is for a very custom qPCR assay. So that requires a whole deal of other things that I have to establish in the lab. So, and we have some cookie-cutter projects or assays, and then other is any custom projects. We're open to any kind of projects to work in the lab. We also support the freezer program. This is located in Moril 1, room N328. There is no shipping fee on this one. It is like a vending machine. There are stocked reagents that can be purchased when the account is created. Also, you can place special orders, and then I coordinate with the user to have it picked up from the facility. My background, I did my PhD in molecular microbiology from Institute of Genomics and Integrative Biology in India. Then I did several years of postdoc at UIC. Then I moved on as a research scientist, and then again I moved on as a research professor before taking this position. My basic research interest is in molecular biology and microbiome research, and also next-gen sequencing. With this, I would like to introduce our next speaker, Elizabeth Rullo, and we are happy to have her here to present the 10x Chromium system for single-cell genomics. Liz, please take over.
Hey, thanks, Ravi. I think there we go. Let me share my screen. Can you see the presentation?
Yep, we
Great,
can.
great, thank you. Thanks, Ravi. Thank you, Andrew, for the introduction, and Lisa for inviting us today. I was telling the group earlier before everyone joined that UMass has a special place in my heart because I did go to undergrad and I got my master's in microbiology from UMass Amherst. So I have spent many, many hours in Morrill, and I feel for any of you who are still in school there, but it was a great experience. And I kind of wish that we could do this in person today just so that I could enjoy some of my favorite meals out in Amherst. But I guess we have to do it the way we're doing it now for a little bit longer. But I'm going to introduce myself. So I am the sales executive for 10x Genomics. And I've got Stephen Wood, who's my colleague, who's the field application scientist. So we're going to kind of tag team. We can do an overview of kind of the core products that 10x has, some of the technology. And then as we get into the Q&A, it's always good to have Stephen here. He's experienced really the hands-on running with customers and some of their projects. And I know there's a few people who are attending today who've actually used the technology. So that helps a little bit. What I'm going to do, though, is kind of give you that basic background to make sure that everyone's on the same page. Hold on a second. Let me see if I can get my slides to advance. There we go. So just a quick overview of 10x. You know, we serve pretty much everybody. Any field, your oncology, your developmental biology, all sorts of model organisms. It doesn't have to be a human sample. And we're in a lot of different universities, core facilities. You know, we do have some service providers, but in general, it's definitely more of an on-site system. The Chromium controller we always talk about, which is basically the system that UMass Amherst would be interested in, is about the size of a toaster. So it's not very large and it's pretty easy to use. So we can run in kind of all sorts of different settings. We have kind of three major themes at 10x that really drive our motivation. And so the first one is resolution, making sure that, you know, we can build tools that really technologies for our customers to use to just get better resolution to their current samples and measure multiple dimensions and simultaneously assess phenotypes to ultimately get to the biological context in your questions. The second is scale. So the more cells, the more tissues, the more samples that you can use, obviously you're going to accelerate your biology. And then the third one is access. So, you know, again, why we're kind of here today is we really want to make sure that you can access the system. We give you the best tools. I think based on Ravi's presentation already, 10x would be a great complement to the current sequencing and you know, DNA and RNA assays that you're currently able to offer. And the key to our technology really in the platform is just the ability to interrogate biological samples at a high resolution, all the way down to that molecular level, right? So traditional bulk RNA sequencing really takes a heterogeneous sample and gets a single data point. So it can mask some of the underlying critical information that's kind of hidden behind, you know, averages and incomplete measurements. So our Chromium platform is fundamental in dissecting tissues and single samples down to the single cell resolution. So we can really uncover transcriptional and chromatin states that are unique to distinct cellular subpopulations. And so while the Chromium platform helps answer the question of what is expressed in the particular sample, our Visium platform addresses the where, and it's our spatially resolved technology. I'm not going to go into a lot about the Visium today. It is one of our newer products. There's a lot of talk, obviously, about spatial technologies and analysis. It's actually basically can be an entire talk in itself, but I will just kind of give you a quick overview so you can see some of the similarities and differences between the Chromium. So, you know, it's not just our marketing slides, but, you know, we've really grown in publications. This is a little outdated. So from 2019, obviously last year, a lot of people were writing and not as much work was being done. So we've really increased the publications and we span a variety of different journals and can really kind of cover any topic. So I always tell customers, if there's something that you're interested in, if you're like, I want to try this, has it ever been done? You can always reach out to us. You can reach out to our tech support team, and they're able to really dive into these publications and see if we've got an experienced user for your particular assay. Knowing that today's group might be just a little diverse, I didn't really want to focus on a particular oncology or a particular immunology topic. But one thing I do want to highlight, because it's obviously near and dear to all of us, is a COVID-19 publication. And this group in China that was able to use, they collected samples from COVID-19 patients, some with severe disease and some with mild, and were able to actually dissect down the individual levels and then compare it against some single-cell RNA-seq data that had been done from a previous group that was healthy patients, right? So non-COVID patients. And you can really see from this graph, this middle group C is actually our UMAP. And these are single cells, right? And you can obviously just, at quick glance, see the difference of clustering and different types of cells that were able to distinguish between the three different groups. And so it really showed that the CD8 T cells affected the mild group contain highly expanded clones. So this is a paper that uses non-malignant gene expression, but one of our other assays, which is our immune profiling panel. So they were able to do some TCR sequencing to really determine the T cell importance, and that in the mild cases, the TCR clonal types had highly and moderately expanded in comparison to the single highly expanded clonal types from patients in the severe group. So that diversity is really important, and that the repertoire of clonal types was important in the response to the viral infection. So as I mentioned in that paper, most people think of us as just a gene expression assay, but what we always like to highlight is that we have a variety of other applications. So our single cell immune profiling, which allows you to do TCR and BCR sequencing. We have a cell surface protein application, so to detect cell surface protein, as well as the gene expression from an individual cell using our feature barcode technology. Visium, as I mentioned, which is our spatial technology. And then one of our assays that was recently launched the end of last year is our multi-omic assay, which is a single cell attack and gene expression out of the same cell. 10x is an innovative company, and we provide a complete solution of reagents, hardware, and software that can easily be incorporated into your current next-gen sequencing workflow. It all starts with a single sample of single cells or single nuclei combined with our reagents and processed on our Chromium controller. Each cell is partitioned into a nanoliter-sized compartment, followed by generating sequencing libraries, which can then be run in the Illumina sequencer. And then we offer Cell Ranger and Loop Browser software that's used for analysis and visualization of the data. So how does it work? One of the key aspects of the technology are these gel beads. So in this cartoon, you can see this individual gel bead here, and it's made up of many oligos, and these oligos have specific features. So for example, in the 3' gene expression bead, we have a poly-VT tail to capture the mRNA. We have a unique molecular identifier for sequencing alignment downstream, and a read-one primer, obviously for sequencing. And then, as you can see here in this green section here, there's a 10x barcode. And so this is a unique 16 nucleotide barcode, and each gel has the same individual barcode, which is used for tagging the cells and later downstream analysis. The second key aspect is the microfluidic chip that's used inside the Chromium controller. So during the workflow, the single cell suspensions combined with master mix and loaded onto the chip, along with the 10x barcoded beads and partitioning oil. The chip is placed in the Chromium controller, where the single cells are partitioned with a single bead into what we call a gem, a gel bead and emulsion particle. So the cDNA synthesis actually occurs inside the gem, and that will uniquely label the cDNA with that individual 10x barcode from each particular bead. And the barcodes, once labeled, the cDNAs can be combined and taken through the standard library preparation and sequenced. And then bioinformatically afterwards, we can distinguish which transcript came from which individual cell based on those initial barcodes. So some of the logistics for running the Chromium controller are listed here. So this is a picture of our chip that's loaded inside the Chromium controller. We load up to 8 samples in parallel. And we target between 500 and 10,000 cells to capture per sample. So a couple of things to consider when you're determining the amount of sample to load, besides obviously what you have available. We have a really high capture rate of about 65% of the input cells are captured into each of these lanes. So ideally, that means you're targeting loading 16,000 cells to yield data from about 10,000. So while most of the gems will contain a single cell, there is a statistical probability that some gels will contain more than one cell. So the expected fraction of droplets with more than one cell increases with the number of targeted cells. So that's why we have some clear suggestions and guidances on how many cells to load for your sample. This process actually takes only about 18 minutes. So as I mentioned before, it's a pretty simple process. You then remove the gems, do some purifications, and go through your standard library prep that most of you are probably used to if you've done any next-gen sequencing. This is a microscopic picture just to show you kind of what it looks like. This is an individual gem. So you can see that we have the gel bead in the middle. We have a single T cell loaded in this. And really within this little gem is kind of almost a PCR machine, right? Your reverse transcription is going to occur here prior to lysing the gems and combining all of the sample together. So one thing that's actually pretty interesting about 10x is, we really wanted to see how we can make this kind of a multi-omic assay, right? So it's not just gene expression, but what other features can we target using the same cell? But we don't want to reinvent the wheel on doing this. So what we've done is actually kind of cool. Instead of having the poly-D-tail oligo that we talked about in the gene expression assay, we can add additional oligos. And in this example, we've got 2 capture sequence oligos. And they still contain that same 10x barcode. So all 3 of these oligos can be in the same single bead and contain the same oligo. But now in this case, we can take these capture sequences, we can make a complementary sequence that can be used to, for example, tag an antibody. So what we're able to do is take that complementary capture sequence, tag an antibody, stain the cells prior to loading them in the controller with this tagged antibody. And then we're able to detect both the protein expression and the mRNA. And again, because that 10x barcode is the same, we're going to be able to pair it together. And so for the antibodies, you can definitely design your own, but we've also partnered with BioLegend to make some pre-tagged antibodies. Some of them are common ones. So it's a good way, again, to really kind of get a multi-omic readout. In addition, we have our single-cell profiling assay. And this kit, again, starts with the same concept. You're going to take your individual cells and load it through, only this time we can sequence from the 5' end, and we're able to capture the TCR and BCR sequences. So, and by combining, if you really wanted to, you could combine this with the feature barcode, which is the protein one we just discussed. And so you could really make it a multi-omic by not only getting the VDJ information, you could get your gene expression, you could get cell surface proteins, antigen specificities, and our other assay that we've really been working on is a CRISPR assay. So we tag the guide RNAs with a complementary sequence to the beads, and so the same process can occur. We're able to detect as much as we can from a single individual cell and map it all back together. So, we can kind of combine all these measurements for a true multi-omic solution. So we can interrogate the cells both at the transcriptional and translational level and truly understand the cellular functions to provide a complete picture. Single cell resolution for gene expression, antigen binding specificity and sequencing and your clonal type frequencies, again, all out of that individual cell and all in a single assay. So it's just a lot of information that you can get from the same system. I know that there'll probably be some sequencing questions here. So to give some guidance, this is kind of just an example. So again, you'll have your single cell initial starting point. You're going to make a TCR library if you're going to do the immune profiling. You're going to develop an mRNA library for pretty much any of the assays that we do, or just a standalone. And then again, we have a feature barcode if you want to do any of that protein or CRISPR assays. The mRNA library, which is, the 3' is 5' probably more popular. We do recommend a minimum of 20,000 read pairs per cell. And so for the other two libraries, these can be a little bit less of a sequencing. If you're not interested in the whole transcriptome, this is one of our newer products. We've actually launched 4 targeted panels. So these panels actually have still over 1,000 genes, but they're going to focus a little bit more on kind of key areas: cancer, immunology, gene signature, and neuroscience. And the process here is the same thing. You'll go through your library prep, but once you generate your library, you're going to take a small amount and you're going to probe it with just the key targets and re-amplify. And so it actually allows you to get high expression of the targets you're interested in, but you can use a less of a, you don't have to sequence as much as you do for the whole transcriptome because you've got very targeted counts. And this is kind of the, overview of the 3' gene expression library. Again, our minimum is 20,000 read pairs per cell and can be run in any of the Illumina sequencers. And the sequencing depth is just really somewhat going to depend on your project and your sample type. Again, we're happy to give some guidance on that. It just really depends on what you're looking to do. Then really fast, just because this is still a fairly new assay, and I know if anyone has done ATAC-seq in the past, it's great to be able to pair this with the gene expression. So this is our only basically DNA assay, right? So we're going to interrogate the DNA here. The chromatin consists of DNA that's wrapped tightly around the histones, and chromatin accessibility can be identified. It can identify regions of the genomes that are available for binding proteins, such as the RNA polymerase and transcription factors, which can really regulate your gene expression. So the ATAC method identifies regions of open chromatin. So we start with nuclei instead of cells, and we incubate with a transposase enzyme, which preferentially cuts any accessible DNA and inserts a tag that has the sequencing adapters. And then the sample is processed through the Chromium controller and downstream sequencing. So the sequencing accessible DNA fragments can then be mapped back to sequencing reads, map the sequencing reads back to the genome to allow one to identify open chromatin regions, including multiple types of genomic features such as promoters, enhancers, and transcription factors. And kind of one of the reasons, again, that we highlight this is that in the past, researchers have had to split their sample and analyze their gene expression and chromatin accessibility separately, but now it can be used, accessed in the same sample. Again, we're going to access the gene expression out of the nuclei sample, but this allows us to really link cell type specific gene expression to nearby chromatin regions using what we call our feature linkage. So these are graphs that come out of our Blue browser. And so you can see, with the blue arc indicating that there's a positive, the positive regulation, and the red arc indicating negative regulation. And these same targets are indicated in the right with your positive and negative gene expression data. So it really allows us to link it together at the individual cell basis. One of the other questions that really comes up, I think I've mentioned this a few times already, is that we really do have kind of an end-to-end workflow. We have amazing technical support, right? So sample prep being one of the most important aspects of 10X. You want nice, healthy individual single cells. And so we have a variety of sample prep webinars and suggestions, demonstrative protocols, technical notes. Again, if you have a sample that you're not sure of, we can always try and find someone who's actually already worked with it. And the same on the software end. Running the actual assay itself is pretty straightforward and very simple, and everyone's pipetted before is able to do it. The software support on the back end is also very good. We've also just launched a cloud analysis, which will help a little bit as well. So for new users, that's a great alternative too. Again, it's we have the analysis tools, visualization, but it is also you are also able to take all the data into any third-party software that you might want to use. And then I wanted to just quickly, in about 2 or 3 slides here, go over Visium spatial gene expression, because it has been such a hot topic lately. Currently, we offer a fresh frozen application, and we are pre-selling our FFPE version, which is going to be really available for shipping next month. So that's why there's been a lot of talk about it lately. Like I said, the chromatin platform is great for uncovering what is expressed in particular tissue, but the Visium platform really addresses the where, and the people with tissue sections are really interested in different cells and where they interact with each other. Kind of the workflow for this is pretty straightforward. So you're going to section your tissue, whether it's FFPE or whether it's fresh frozen. And we section them onto our Visium library prep slides. You visualize them either with an H&E or immunofluorescent stain. The cDNA synthesis actually occurs on this slide. And so after that cDNA synthesis occurs, we do a standard gene expression library prep, sequencing, and then we have our analysis, which not only takes into account your gene expression data, but also integrates into it with your immunofluorescence or your H&E stains that you've taken. And the way this works is, again, not really reinventing the wheel, but instead of, you'll notice over here, we've still got this individual oligo. What we've got are these Visium slides with 4 distinct gene expression sections. And each of these contain about 5,000 barcoded spots. And the barcodes are about 55 microns in diameter. And within each of these spots is a lawn of these oligos. So instead of using your bead this time, it's actually a spot on the slide. And again, the 10x barcode here is going to reference back what spot on the slide that sample came from. So after the fresh frozen tissue sections are placed on the slide, right? The samples are permeabilized into the RNAs released and hybridizes to the poly-D tail here. The cDNA synthesis occurs right on that slide. Then again, they're all mRNAs, the cDNA is all barcoded, and so it can actually be pulled together. And it gives some pretty cool pictures. So this is an H&E slide as well of a mouse brain, and then we've got 2 hippocampus genes. Then you can see here, keeping in mind that these are heat maps; these are not fluorescently stained slides. So the heat map indicating the high expression of the particular genes, and just like we do for our single cell analysis, where we can do it down to the individual gene level, we can also do clustering and things like that with the Visium expression as well. And with that, I will thank you and see if anyone has any questions. We want to make sure we leave some time.
Yes, I have a few questions. Thanks for the presentation and capabilities that for the chromatin controller. So is there any way where we can know like what was the capture rate, like what is singlet, doublet, how do we, after the gel bead and the emulsion, how do we know what was the capture rate?
Right, you will get a readout of the number of cells that it, right, the software will show you the number of cells that it's analyzing.
But how does, whether we captured a single cell or it is doublet during the process or when we put the sample, when you put the cells on the controller?
So you will have an expected doublet rate just based on the number of cells that you're targeting. Because you're loading it, it's governed by a Poisson distribution in the microfluidics. So you'll know based on the number of cells that you're targeting what the estimated doublet rate would be. Our Cell Ranger pipeline doesn't have the ability to identify or filter those out based on just high UMI expression, but it is possible to use a number of third-party tools to look for high UMI expression, and then where you have multiple profiles of 2 cells being present for the same barcode, you can use that to discriminate and then filter that out of the data.
I see. Okay. Could you also speak more about the CRISPR? You mentioned that now we can do some CRISPR assays on using the controller. Could you elaborate on that a bit?
Yes.
Sure. Oh, go ahead, Steve.
So you can generate your own, you can generate your guide RNA to have homology to those 2 capture sequences, either the capture sequence 1 or the capture sequence 2 that are present in the 3' gene expression assay. So those gel beads have those 2 capture sequences, so you can directly pull down those guide RNAs and look at the transcriptional profiles for whatever those guide RNAs are targeting. So that way you're directly pairing whatever your CRISPR system is inactivating, or if it's CRISPR activation, you can have that direct association of your guide RNA with the transcriptional profile of those cells.
And we worked a little bit with, we're working with Millipore Sigma. So they're helping design some of the vectors. They've got some of kind of the optimization kits for people to try, and they will actually do as much or as little of the work as you want in that kind of upstream guide design.
So what is the average, let's say for the gene expression assays, what is the range for cost per cell? I know that when we have shown that you can capture up to from 500 up to 10,000 cells, so that's going to affect the cost, right? So what is, let's say, like if I were to say that, like it is going to cost $1 per cell, or
right? It's about $1,500 per lane. So that's the fixed cost, and then depending on the number of cells you choose to recover from that.
No, but let's say if I were to do the entire costing, for example, let's say with sequencing and everything, is it safe for us to say that it is 50 cents per cell? That includes, sequencing cost also.
So I can't factor in the sequencing cost, unfortunately, because every institution has different, right? So on the 10x side, if you were doing, recovering 10,000 cells, it's or if you're recovering 100, like the price per assay is going to be the same, right? So it's still, each sample is going to be 1,500, and then the cost of the sequencing.
And for the data analysis, I know that 10x is a very good support for the data analysis in terms of processing as well as visualization. How about for non-model organisms? What support do you have for non-model organisms?
So a lot of that, it really comes down to the quality of the reference that you're going to be using for the model organism. So some are going to be much better than others. So that will definitely impact the overall quality of the mapping that you're going to have. The whole process, though, it's going to be the same, whether it's human, mouse, zebrafish, whatever organism you're working with. You're still going to use the same software. So you can, if you have a reference that you have available, you can generate that reference for Cell Ranger and perform the assay or perform the analysis using that software accordingly.
I know that before the Chromium was able to support the high molecular weight DNA sequencing. Is there any options with a newer system or the newer workflow?
Yeah, unfortunately, that product has been sunset, and right now there's no plans to bring that back. So,
I have one last question, and so if you were to consider this the Visium for spatial transcriptomics, what resources do we need? Obviously we will require the system from 10x, but what other things we may need?
It actually doesn't require a system from us. This is a little different than the chromium, but it does require a histologist who can section. So a group that's got, any experience in FFP or fresh frozen, and then some imaging capabilities that will allow you to make sure it can image the sample. But the actual 10x part of it in the molecular biology is really it's going to occur on that slide. And so all there you need is your thermal cycler and some basic library prep reagents for that.
Liz, we have a question in the chat. Sorry, Rob.
No, please, yeah, I was about to say that, so go ahead.
Okay. Jesse asks, can you comment on coverage and abundant versus low number transcripts from only 20,000 reads cell?
So I can take that one. It really is going to depend on the expression level of your sample, whether 20,000 read pairs, reads per cell is going to be a sufficient sequencing depth for your sample. For many cell types, that might be sufficient, but for other cell types like a primary cell line, primary cells, they often require much higher sequencing depth in order to reach a reasonable saturation of your data, where you're able to pull out those transcripts that you're interested in finding. So if it's something where you have a very high expressing cell, then you might need to consider going to a much higher sequencing depth of 50, 75, or even 100 reads per cell.
All right, Kai, can I expand on that a little bit with you?
Yeah.
So where do you suggest someone starts? So I work in early embryo, so maybe no one's ever done this before, this approach on these samples. Where do we start figuring out read depth and how?
So the first place I would start is just, sure. The first place is just to go to the publications and see if anyone has done your cell type of interest and see if that's been looked at, just to get a sense of where people in the field have previously sequenced, what depth they've looked at. If you don't have that information and you don't have a sense of what would be a reasonable cell depth or sequencing depth, what I recommend is that first experiment that you do, sequence at a very high sequencing depth, try 75 to 100,000 reads per cell. Do your complete analysis on your data at that high sequencing depth, and then you can downsample and do your analysis at lower sequencing depth, just downsampling that data, and see whether you're able to recapitulate that same information that you get from a high sequencing depth. That way you know that you're not losing any information if you were to go for future experiments to sequence at a lower depth. But that way you also know that you're not just wasting sequencing by getting reads that are not going to be useful for the questions that you're asking.
And how many cells in that scenario do you start with?
So on a particular channel, so you can run 8 channels in parallel. You can run up to 10,000 cells on a single channel. So you could run up to 80,000 cells on that chip. Whether you want to run all 10,000 really comes down to whether you have a very homogeneous population or if it's rather heterogeneous and there's a rare population that you think you're going to be able to pull out. So if it's got a more rare population there that you're interested in, you might need to go towards that full 10,000 cells on a channel in order to capture those more rare populations. If, however, you have a rather homogeneous population, you might not need to have more than 2,000, 5,000 cells. It really comes down to what the question is that you're looking at, what the rarity of that population is, and also considering what the sequencing depth is going to be for those samples, because obviously as you increase the number of cells, you're also going to increase the amount of sequencing that's required.
Great, thanks.
Yeah.
The other thing to consider is we keep talking about the 8 channels. There's many customers that run 1 or 2 samples, right? So these are all freeze thawable reagents. So you can run 1 or 2 samples at a time. You don't have to make that full commitment to 8 channels your first run.
Can I ask about how does the quality of the sample, I mean, how viable the cells are, or things like that, how does that impact the quality of your result?
Yeah, that's a great question. And sample quality is a really important aspect of any single cell experiment, especially with droplet microfluidics, because if you have dead or dying cells, for one thing, those are more likely to rupture and break open and release a lot of debris. That debris can result in clogs in the microfluidics, and you actually will have a failed run. So you won't generate the emulsion to be able to generate your single cell libraries. The other thing to be mindful of is that when you have a lot of ambient RNA, that ambient RNA is going to be picked up in the droplets and barcoded along with your intact cells. And so that's going to show up as just background noise in the transcriptional profile. So ideally, we say that you should target around 90% viability in your sample. For some samples, that's feasible. Others, it might be a lot more challenging. You're going to have to do some more processing to remove dead and dying cells, maybe do an enrichment step through flow cytometry or flow sorting. There's different ways that you can handle your sample to enrich to get a more viable sample. But ideally, you do want to target a very high viability sample so that you can get the best quality data out of the assay.
Would it be possible to sort for dividing cells with some kind of, membrane dye like CFSE, or do you know if anybody has done that?
That's something I would have to look into just a little bit. Theoretically, that should be feasible, but there's some things I would want to look into, just the impact that a dye like CFSE might have on any of the transcripts. Off the top of my head, I can't tell you yes, but I think that there should be ways that you could specifically enrich for those populations with some markers that you can run them.
And is there anything that you can put your cells in so that they, sort of guards the RNA, like RNazole or not RNazole, sorry, RNA later?
Yeah, so really the goal is to work as quickly as possible from going from tissue or whatever state your cells are starting with to running them on the controller.
Mm-hmm.
So you can either do that where you are taking your sample and dissociating that sample into a single cell state, and then you can freeze that down and come back until you've collected your samples and then process everything together, or you can process them in batches and then carry them through when they're in a fresh state. There are ways that you can use, you can do methanol fixation, but that does also have an impact on the transcriptional profiles. So it's feasible, but there is a downside to that as well. It really depends on whether you need super high fidelity in the data. It depends on the samples that you're working with. So there's a lot of things to take into account when you consider the sample preparation, and sample preparation really ends up being really the thing that you end up spending the most amount of time trying to optimize to get as high a quality as possible. Unfortunately, though, there aren't like ways that you can just sort of keep your cells intact at the moment. We do have a fixation product that's going to be released in the second half of this year. I don't have any details though on that or the performance that you will see with that.
Can I also ask about the TCR biased or emphasis that you make a separate library is what you're saying? It's not on the sequencing level, it's on the library construction?
Right, so you're going to collect the entire cDNA. So if you use our 5' product, it allows you to capture your full-length VDJ sequences. And so the VDJ transcripts are going to be present in your cDNA. What we do is we'll take the cDNA and carry that forward for our gene expression library. We're also going to take a portion of that cDNA and do a specific enrichment step using the constant regions for those TCRs. And so what that allows us to do is specifically enrich for TCR sequences and prepare those separately as a separate library. And so we can use a separate sample index and then sequence that all together so that we can link together the transcriptional profile as well as those TCRs. And so we're able to computationally build out the full length of that TCR, going from an NGS library to a full length 600 to 700 base pair contig sequence of those VDJ sequences. And so we're capturing all of those VDJ regions as well as the CD3 region, CDR3 region. And so we can look at TRAs and TRBs to look at those samples. Additionally, with that, you can also use our decode dextramers. So Immudex is a partnering company that generates dextramers. So they're very similar to tetramers, they just have a longer moiety to them. And so you can use that to look at antigen specificity. So if there's an antigen that you know that you want to look at, you can build those dextramers, stain your cells, and so you're able to look at antigen specificity, the TCR sequences, as well as the gene expression, all from the same cell.
So if I wanted to look at, say, the gamma delta TCR in a different species, though.
So yeah, for the specific enrichment sets that we have that for human and mouse, for alpha and beta. That being said, there are a number of people that have generated their own specific sequences for gamma delta T cells.
So you can
So that's something that you could certainly do. And if it is going to be for a different species, you could also do it. So I'm aware of people that are doing in primate research, they're looking at gamma delta. So you can generate those specific sequences. So it either might have already been done by someone and you can just find the primers that work. Otherwise, we can work with you to identify what will be optimal in those situations.
Okay, thank you.
Yeah. Liz, did you get all of the?
I think I got all the questions in the, yep. So again, it's one controller that does all of the different assays, right? So it's just different kits. And Visium doesn't use an instrument.
There's one question that is about the cost.
Yep, the cost of it. So the attack and the gene expression, so our multi-omic kit is about $2,500 per sample. So you add about $1,000 because it's basically two assays in one.
So the capture area is 6.5 by 6.5, 6.5 millimeters. And there's 4 of those capture areas then on the gene expression slide.
So what do we need for the spatial transcription? Do we need to buy slides, or I mean?
Exactly. Yeah, you buy the slides, and that way everything's done, and we sell them in packages. You know, you can buy a single slide or packages of 4 slides.
So what is the cost for the slides then?
That's actually again, it's going to be about $1,500 per sample.
So basically, we can then prepare a section. I mean, one tissue or one slide, basically, one section.
4 sections per slide. So it depends on if you want to do 4 total different samples or if you want to do 4 sections of the same sample. It kind of depends on the work, the researcher's work.
This is like a targeted assay, or this is going to be like?
It's not, no. So the fresh frozen one, you're just going to permeabilize. So all the poly A tails will attach the oligo DTs, right? And our FFP, we have a little bit of a different chemistry because obviously those are very degraded samples. We do have a 2-probe hybridization that then ligates, but it's 18,000 genes. We have human and mouse and 20,000 for mouse. So it's not it's considered a whole transcriptome, although it's with targeted panels.
So then after the slides, we stain the slides, and then we have to, stain meaning, sorry, we have to add the tags, and then we have to prepare the library and sequence it. Is that the workflow?
Correct, exactly. Yeah, you're going to stain the slides, take a picture, and then prepare the library and sequence.
So for the picture, do we need like a high resolution microscope, or?
You need a microscope that has a scanning stage, and it depends on whether you're going to be doing either H&E staining or if you're planning to do immunofluorescence with it. And so there's going to be different imaging requirements. I mean, obviously you'll need an epifluorescent scope or a scope that has a color camera if you're doing H&E.
This is going to be across multiple cores. Or like one for imaging, one for sequencing, and
Exactly. It's, we've done it for a couple institutions now, so we're definitely getting that experience of how to work for each individual core and where the stopping points are to really help you so that, you don't have to do the entire process in a single day. Who can take and, put it in the freezer for a week or two before you do the next step. And, again, wanted to mention it because obviously it's a very popular topic right now. We're doing a lot of 10X has got a lot of webinars on it. There's just a lot of buzz about it, especially because the FFP is new and there's been a lot of translational researchers who've been waiting for it for clinical samples. So make sure you guys know that it's kind of fun. We're, again, always trying to evolve the technology, come up with new products. We do want to keep our basic single cell reagents and the Chromium controller, what other things we can get out of there and what other assays we can run on that. The controller is really key too.
So one question for the cell quality. How about the cell size distribution? If we have like a different size range, different sizes of the cell in the same sample, does it have an effect, or it has to be like the cell size to be uniform? How will that affect the gel bead, the gel emulsion bead?
Sorry, can you repeat? I was responding to a different question. So
the question is, how about the cell size? Like if you have in a sample, if you have different cell size, let's say, 5 micron, 10 micron, 15 micron, is a mix of cells with sizes in the same sample. Will it affect the after the chromium, will it affect the workflow? Or
no. It really won't have an impact. The one thing to do be considerate of, though, is just that the upper limit of what we recommend for sample size is 30 microns. And that's due to the fact that the microfluidic channel itself is about 50 microns. So higher than that, you run the risk of getting clogs. So in those situations where you're going, bumping up into that size, we would recommend that you would run nuclei as opposed to whole cell. Other cases where you're working with like cardiomyocytes or neuronal cells that have long processes, those samples are also challenging to run as whole cells just because they have a tendency to clump up and are not as pliable to go through the microfluidic channel. But having a rather heterogeneous size distribution, as long as it's under that 30 microns, that shouldn't be an issue.
Do you have like a lower cutoff too? Like a lower cutoff for the cell size?
No, I mean, like I said, you can run nuclei and those work just fine.
So are there any other questions? It's 3 o'clock. I don't want to keep people waiting too much longer. So I want to thank Liz and Steve. That was really informative. And thank you so much, Ravi. And thank everyone for attending today's seminar. This was the last seminar for this spring semester. And as Andrew previously mentioned, we'll be offering a few special seminars over the summer. So stay tuned. Goodbye, everybody.
Thank you.
Thank you. Bye-bye.
Bye.